Alright guys, so we left off talking about ribosomes, both free and bound. We're going to pick up here with a new topic. So this topic is the endomembrane system and how that system regulates proteins and performs metabolic functions. Metabolic refers to any physical or chemical process that takes place within the cell. The first organelle we're going to talk about today is the endoplasmic reticulum. That is this light blue shape that you see over here. The dark blue is the nucleus. This endoplasmic reticulum makes up more than half of the membrane structures that are found within many cells. It's a network of both membranes and sacs. So if you look over here at the light blue picture, the kind of empty space, or what appears empty, that's the sacs they're referring to, and those sacs are called the cisternal space. I told you guys before that the nucleus is continuous with the endoplasmic reticulum. You can see that here. So here you can see where one stops, the other begins. And then the endoplasmic reticulum is wrapped around the nucleus. There's two types of endoplasmic reticulum. So we have the smooth and the rough. Both of them have different functions. So we're going to talk about the smooth ER first. So in the smooth ER, we have the synthesis of lipids. And synthesis means to create, so the creation of lipids, the metabolism of carbohydrates, and the detoxification of drugs and poisons. So the smooth ER performs all three of these functions. Then we have the rough ER, and it's really easy to recognize because if you look at the picture, the rough ER has ribosomes all over it. That's these little orange dots, and the smooth ER does not. So if you were looking under a microscope, the rough ER would actually appear to have a rough texture. The ribosomes on the ER synthesize or create proteins, and those proteins are then secreted by the cell. As those proteins are produced, the polypeptide chains travel across the endoplasmic reticulum membrane and into the cisternal space. And within the cisternal space, those proteins are then packaged into transport vesicles, which bud off the ER and move towards the Golgi apparatus. So that was kind of confusing. So here's a picture of that taking place. So here we have the ribosomes. Remember, ribosomes are where proteins are created. So we have this polypeptide chain here. Polypeptides are monomers, remember, of proteins. So we learned about that when we talked about chemistry of life. So here, this polypeptide chain is located within the cisternal space. That's this big empty space here. And then once it's ready for transport, a bud actually is created where it breaks off of the endoplasmic reticulum, and it is now in a transport vesicle for shipment to the next organelle, which is the Golgi. This is the Golgi apparatus, so it operates similar to a post office. So this is where the proteins come in from the endoplasmic reticulum. This is the structure of the Golgi, so it actually has polarity to it. So this smaller section here is known as the cis face, whereas this larger, more broad section is known as the trans face. Well, the cis face is where those transport vesicles come in. So they leave the ER, they come to the cis face of the Golgi, and then inside this area here, that's where they're modified, stored, and then shipped out. And when they're ready for shipment, they leave out the trans face and these secretory vesicles that you see here. This has a very similar structure to the endoplasmic reticulum. It also has those sac-like spaces that are known as cisternae. It has its own membrane that you can see on the outside. And just keep in mind, remember that the incoming ones come to the cis side and the exiting ones leave out the trans side. Our next organelle is the lysosome. So it is also membrane bound. So you can see in the picture there's a membrane on the outside. The inside is full of enzymes. Those enzymes are actually called hydrolytic, and that's because they participate in hydrolysis. And if you remember, hydrolysis is the breaking apart of the large macromolecules. When those large macromolecules are broken apart, you have their monomers. And then those monomers are released back into the cytosol and reused by the cell. We talked about how enzymes work best at a particular pH. So inside the lysosome, it's an acidic environment, and the enzymes work properly here. Well, if that lysosome was to break open or leak, that enzyme is not going to be very active because now the pH has changed. So inside the lysosome, it's very acidic, but outside it is not. So this is another reason why it's really important for organelles to be compartmentalized and separated by a membrane. Vacuoles are our next organelle. They're once again membrane-bound vesicles. And here we've got two different examples. So this one shows endocytosis. So we have our lysosome with particles on the outside, but the lysosome wants to take in that particle, so it moves closer, and it actually conforms its membrane around the particle that it wants, pulls it inside, and then digests it. The contractile vacuole is, this example shows water. So basically water moves into the vacuole until it's full. As the vacuole contracts, the water is expelled, and then it can take in more water. The central vacuole is the one you're probably most familiar with. This one is found in plant cells, and it's one of the biggest differences between plant cells and animal cells, and it can take up to 80% of the cell's space. So as you can see, it's very large. We're going to move on to the next topic, which is mitochondria and chloroplast change energy from one form to another. So these are how the different cells get their energy. Mitochondria is where cellular respiration occurs, and cellular respiration is the process that uses oxygen to create ATP, and it does this by extracting energy from sugars, fats, and other sources. This is found in both plant and animal cells, and it has an outer and inner membrane. So if you look over at the picture, the outer membrane is that dark orange color, the inner membrane is the yellow section. And then the inner membrane is folded to increase the surface area. So we've been talking a lot about surface area in class. This is another way to increase the surface area for the mitochondria, and that enhances cellular respiration. It has these folds in it, once again, that are known as cristae, very similar to both the endoplasmic reticulum and the Golgi. This inner compartment is the mitochondrial matrix. It is fluid filled, and there's actually DNA floating around in here in ribosomes. So we all know already that DNA is located inside the nucleus, but mitochondria actually has its own set of DNA and its own set of ribosomes. The chloroplasts are found in plants and algae. These are for organisms that cannot make their own food. It's the site of photosynthesis. And there's a theory that exists about mitochondria and chloroplasts. So scientists believe that mitochondria and chloroplasts have a similar origin. The theory states that these organelles, so the mitochondria and chloroplasts, descended from prokaryotic cells that were once engulfed by the ancestors of eukaryotic cells. So what that means is they think a long time ago there were eukaryotic cells, and the mitochondria and chloroplasts were actually prokaryotic cells. And a long time ago, those eukaryotic cells took the mitochondria and chloroplasts in. And we have some evidence for that. So these are 3 pieces of major evidence. So the first one, both organelles have a double membrane. Both organelles have their own ribosomes and DNA. And both of these organelles reproduce independently within the cell. Our next organelle is called the peroxisomes. It has a single membrane, so you can see that in the picture over here. It performs various functions. So one of these functions involves transferring hydrogen from other compounds to oxygen and producing hydrogen peroxide. It can also break down fatty acids that are then sent to the mitochondria for fuel. And they can detoxify alcohol by transferring hydrogen from poison to oxygen. Our next topic is the cytoskeleton. The cytoskeleton is a network of protein fibers that run through the cytoplasm. So this is a picture of a piece of a cell. And the cytoskeleton is responsible for support, motility or movement, and then regulating some biochemical activities. And we have three types of fibers. So we have the intermediate filaments, which you see here, the microtubules, and the microfilaments. The first one we're going to talk about is microtubules. So that is this one right here. It is made out of the protein tubulin. It is the largest of all three of the fibers, and it provides shape and support for the cell. It also can serve as tracks. So we have motor molecules, which are molecules that have movement, and it can provide the tracks for which these molecules move. It separates chromosomes when we do cell division, like mitosis and meiosis, and it actually forms the spindle fibers. And it is the structural component of both cilia and flagella, which we'll talk a little bit more here in a few minutes. The second one is microfilaments. So that is this one right here. This one is made out of the protein actin. So remember the one before was made out of tubulin. Now we have the protein actin. It is smaller than the microtubules, and it functions on a smaller scale for support. It can be coupled with a motor molecule, so a molecule that moves called myosin, and it can aid in movement. So we have two examples here. Amoeboid movement. So if you've ever seen an amoeba, it kind of looks like it's crawling. And then we have contraction of muscle cells. And our last one we're going to talk about is the intermediate filament. That is this one right here. And it's slightly larger than microfilaments, but smaller than microtubules. So it's somewhere in the middle, and it's more permanent. It works to maintain the shape of the cell and fixing the position of some organelles. Now we have centrosomes and centrioles. So they're very similar. Centrosomes are located near the nucleus, and this is where microtubules grow. They can also be referred to as the microtubule organizing center. And in animal cells, centrosomes contain centrioles. Centrioles replicate before cell division, and they have a special arrangement responsible for the beating movement of flagella and cilia. So this over here is a picture of a centriole structure. Flagella and cilia are basically, they're parts of the cell used for movement. So the flagella is in this picture right here. They're very long, so much longer than cilia, and few in number. Typically there's maybe one or two. Unicellular eukaryotic organisms are propelled through the water by flagella, so used for movement. And some examples where you could find flagella would be in the sperm of animals, algae, and then some plants. Cilia is this one here on the right. It's much shorter, used for movement as well. When stationary, so when it's attached to a tissue, they can actually move fluid over the surface of that tissue. And an example would be the lining of the trachea. So in your throat, when mucus is trapped, the cilia helps move the fluid along the surface. So they also have different movements. So here the flagella works more like a propeller, and then the cilia beats back and forth. So even though they have a different number and they're different in length, they have a common structure. So this is the structure that they share. So they're composed of microtubules, remember. So typically there's 9 pairs of microtubules on the outside, and then there's a central core that has 2, and then this is referred to as the 9+2 pattern. The next topic is extracellular components and the connections between cells that help coordinate the cell's activities. So the first thing we're going to talk about is the cell wall. So the cell wall in plants provides protection and helps maintain the cell's shape. It's outside the cell membrane. So if you look in this picture, the cell wall is the outermost layer of the plant cell. It's primarily made out of cellulose. And then there are some other cell types that also have